Hello everyone. First, I want to thank RNA 2023 for giving me this opportunity. My name is Zhe Zhang. I'm a postdoc from Shenzhen Bay Laboratory. Today, I'm going to share our previous work on lantern self-cleaving ribozymes. Self-cleaving ribozyme is a type of non-coding RNA which can catalyze the site-specific cleavage of its own phosphodiester backbone. So far, there are only 10 classes of self-cleaving ribozymes. In this presentation, I'm going to talk about the identification of the new ribozyme class, lantern ribozyme. In our previous work, we accurately inferred the full base pairing structures of two self-cleaving ribozymes by using a combination of deep mutational scanning and covariation analysis. One is the HBV-like CTB3 ribozyme. The other one is the crystal ribozyme. In the deep mutational scanning experiment, first we generated the mutant library by random mutagenesis. Then, after high-throughput sequencing, we calculated the relative activity of each mutant by counting the sequencing reads of cleaved and uncleaved RNAs. After that, we used support vector regression and the mutation-independent model to predict the relative activity of the double mutants. The difference between predicted relative activity and observed relative activity can be assessed by covariation-induced deviation of activity, that is CODA. The larger deviation indicates that the two positions in double mutants are more likely paired in the structure. Here we apply the same strategy to two previously proposed self-cleaving ribozymes. One is located at the reverse strand of the olfactory receptor OR4K15 gene. The other one is located at the 5' UTR of an ancient LIE1 transposon. They were discovered along with the well-known CTB3 ribozyme from a science paper. However, little was known about these two ribozymes. By using deep mutational scanning, we found that only a small part of the LIE1 ribozyme was sensitive to mutations. This suggested that only this region was fundamental to the structure and function of this ribozyme. And according to the covariation analysis result, we can see that the structure of the functional region is very simple, only made of two stems and two internal loops. And we observed a similar relative activity distribution and secondary structure in the OR4K15 ribozyme as well. According to the distribution of the relative activity, we further validated the base pairing structure by deep mutational scanning of the functional region. And all the results indicate that the functional regions of these two ribozymes adopt a similar secondary structure that's shaped like Chinese lantern. That is why we call them lantern ribozymes. In biochemical experiments, we confirmed that both two lantern ribozymes were able to cleave after removing the external stem loop. And they employed the same internal phosphodiester transport mechanism for cleavage as the analog RNAs containing DC were not able to cleave. A total loss of cleavage activity in presence of cobalt hexamine chloride indicates that the lantern ribozymes require the binding of divalent cations, not only for structure, but also for catalysis. What is interesting is that the secondary structure of both two lantern ribozymes are circular permutated with two nearly identical internal loops. And the internal loops in lantern ribozymes show a high similarity with two previously reported twistor-sister ribozyme structures. One is the four-way junctional twistor-sister ribozyme. The other one is the three-way junctional twistor-sister ribozyme. Based on the crystal structure of the more identical four-way junctional twistor-sister ribozyme, we generated the homology models of the two lantern ribozymes. According to the model structure, both two stem regions are extended by the additional base pairs in the internal loop region. However, due to the lacking of long-range interactions in twistor-sister ribozymes, U38 in lantern ribozyme is not that conserved, as we found two mutations with medium cleavage activities. And the stable square array of hydrogen bonds found in twistor-sister ribozyme is not that conserved in lantern ribozyme as well, as we found next mutation, A39U has a relative activity as high as 0.8. We also investigated the two nucleotides at the cleavage site, cytosine and adenine. In twistor-sister ribozyme, the cytosine is not sensitive to mutations, while the adenine showed a high sensitivity to mutations. But the mutation effects in lantern ribozymes are different. Those differences suggest that lantern ribozyme may not be simply a minimal version of the twistor-sister ribozymes. A detailed explanation for this difference may require high-resolution structure determination of the lantern ribozymes in the future. Here is a summary of this work. First, our work has shown that different mutational scanning can be used to identify the functional regions of RNAs. Second, like the co-evolutionary information used in AlphaFold 2, covariations in different mutational scanning can also be used to get a nearly full base pairing information of RNAs. Third, we identified a new ribozyme class called lantern ribozymes with proposed 3D structures from human genome. In the end, I would like to express my gratitude to my postdoc supervisors, Jiaqi and Haiyan, and the collaborators Jian, Peng, and Xu. And I also would like to thank the China Postdoctoral Science Foundation, Guangzhou RNA Club, and Shenzhen Bay Laboratory for their support. Thank you.